Hey everyone, it's Katherine, a 4th grade TA at the Young Scientists Program through USC. I normally teach at Norwood and Mack Elementary Schools. It's our last week of science, so I hope you're super excited to learn about viruses this week. Our agenda for today is as follows. We're going to learn about what a virus is, as well as differentiate DNA and RNA and look at the viral structure. We're also going to talk a little bit about COVID-19. Our prediction is going to be about how viruses infect healthy cells. We're going to do a virus model for the experiment and end with our conclusion. Write this in your scientific journal. Our question box for today is, what is a virus? Before we talk about viruses, we're going to learn about DNA and RNA. DNA is deoxyribonucleic acid, and it's a two-stranded genetic material. It has thymine paired with adenine, which are two of the nucleic bases. However, in RNA, which is ribonucleic acid, it's only one-stranded. And instead of thymine, there's uracil. While DNA keeps genetic material safe in our cells, RNA serves as an intermediate between DNA and proteins that our cells need to function properly. But in viruses, RNA takes the place of DNA and is used as the genetic material that infects healthy cells in our body once a virus has entered our body. COVID-19 is a type of virus that comes from the SARS coronavirus family. SARS stands for Severe Acute Respiratory System. Therefore, the virus typically settles in our respiratory system, such as our nose, mouth, and throat. Because coronavirus is a type of virus, we cannot use antibiotics to treat ourselves because it is not a bacterial infection. In fact, there is not really a treatment that gets rid of the virus. Instead, we use vaccines to insert a part of the virus that may not be as strong or as complete, so that our body can learn to make antibodies which will fight against the virus by itself. Now that we've learned a little bit about viruses, let's fill in our prediction. Finish the following sentence. Most viruses infect healthy cells by blank. Now it's time to build our virus model. These are the materials you're going to need. A styrofoam ball of any size, multicolored or same-colored push pins, and yarn. Yarn is not required, but we can use it to represent RNA. You might also want to grab scissors to cut your yarn. To start, take your styrofoam ball and your push pins. One by one, start attaching your push pins into your styrofoam ball. The styrofoam ball, in this case, represents the protein shell of the virus, and the push pins represent the receptors that the virus uses to latch onto and infect a healthy host cell. Be careful, try not to pin yourself. Your styrofoam ball will ideally look a little bit like this picture. Now you can tie your string of yarn to one of the push pins, or use tape to attach it directly to the styrofoam ball to represent the virus's genetic material entering the host cell. If you decide to add yarn to every push pin, it should look a little bit like this. In your observation section of your notebook, describe how you created your virus model. Now it's time for your conclusion. Fill in the following sentence. I learned that viruses blank. Here are a few more questions that you can add to your conclusion if you want to talk a little bit more about your virus model. Number 1, how do you think you could improve your viral model so that it is more realistic? Number 2, do you think all viruses have a similar structure? What aspects of their structure might be different? Now it's time for our Scientist of the Week. Anika Chebrolu is a 14-year-old student from Texas who just won the 2020 Young Scientists Challenge for a discovery that could provide potential therapy to COVID-19. Her amazing research and invention uses in-silico methodology to discover a lead molecule that can selectively bind to the spike protein of COVID-19. She wants to become a doctor or researcher when she grows up. It's incredible to hear about the achievements of young students. It really tells you that you don't need to wait to finish college and become a full-time researcher before you can start doing incredible things with science. Thanks for participating in this week's lesson. I hope you guys enjoyed it. I had a really fun time making videos and teaching science this school year. I hope the rest of your year goes really well, and I hope you have a really fun summer.